All right, welcome to 3.3 review on meiosis. Again, a big thank you to Chris Payne at Bionology for these fantastic presentations. So we're going to be looking at the highlighted statements as per your request, U2, 3, 4, and 5, and U7, as well as A3 and S1. Okay, U1. One diploid nucleus divides by meiosis to produce 4 haploid nuclei. Okay, so this goes back to the fact that meiosis is a reduction division, or a reductive process, where we're starting with one that has a diploid number of chromosomes, right? So a diploid body cell, which has homologous pairs of each chromosomes, except for the sex cells, and the chromosomes are going to be replicated to form sister chromatids in interphase. You're going to get some crossing over in prophase 1, and then metaphase 1. You have the first division of the nucleus here, up through telophase 1, and then you can see each of those are going to go through their second division of the nucleus, where it takes each of the chromosomes and it separates the sister chromatids to end up with 4 haploid gametes that contain one of each chromosome. Okay, and that's really important because they need to be haploid, so that when one haploid gamete combines with another haploid gamete, you end up with a 2N zygote. And so through meiosis, you reduce the number, the amount of information by half with one copy of each chromosome. To continue this, there are a handful of different step-by-step animations and things like that that you could certainly go through, and I will put the link to these this presentation so that you could go and click on them if you'd like. They're a really great way to visualize. You can certainly watch them and take some notes on it. Okay, U6 and U7 review really quickly from topic 3.2. You can pause it and you can look back over this, but you do need to do a quick review before we go to the next slide on diploid nuclei having pairs of homologous chromosomes, and that haploid nuclei have only one copy of each chromosome. Okay, 3.3 U2 is talking about the halving of the chromosome number to allow a sexual life cycle with the fusion of gametes. If you don't need to review this, you can certainly just stop listening for a minute. But let me just talk to you about this really quick. So many eukaryotes are going to reproduce via sexual reproduction. So even organisms that can reproduce asexually will reproduce asexually. I'm sorry, some that can reproduce asexually will reproduce sexually as well. Sexual reproduction involves fertilization. Fertilization is the fusion of gametes or sex cells, one from each parent. This is true of plants. This is true of most animals. This is true of, fungus and all sorts of cool things. Okay, so this happens all the time. Fertilization is going on all the time. And because fertilization involves the fusion of gametes, the number of chromosomes in the next generation is going to be doubled, right? The gametes, if they weren't halved, you'd end up with twice as much genetic information in the next generation. So to prevent that from happening, each generation goes through this halving mechanism during the life cycle. And this is done during meiosis, right? So to compensate for that chromosome doubling during the fertilization of gametes, what happens is they go through meiosis, which halves the chromosomes so that the gametes have half the genetic information before they fuse via fertilization. Okay, that's U2. Here's U3. DNA is replicated before meiosis so that all chromosomes consist of 2 sister chromatids. So in the S phase of interphase, before meiosis ever happens, DNA replication takes place. What happens here is that the chromosomes are going to be replicated, and these copies are going to attach to each other at the centromere. The attached chromosome and its copy are known as sister chromatids. So this is the sister chromatid of this. This is the sister chromatid of this. After the S phase, you're going to get more growth and preparation for meiosis. So this pair of sister chromatids and this pair of sister chromatids are a homologous pair. Because say this is chromosome 1, this is also chromosome 1, but maybe this is the inherited copy from dad and this is the one from mom. But during the S phase, they are going to each duplicate, as with chromosome 2. All right, U7 is about crossing over. So crossing over and random orientation promotes genetic variation. That's what U7 says. So there's a lot of content on here. You can pause it if you want and kind of read over the text if you need to. So during prophase 1, you can kind of see that this DNA gets really close to each other in homologous pairs, right? And that crossing over is where there is an exchange of genetic information, where a handful of genes will actually swap places, okay? And this ends up with a recombination of alleles, right? So that's crossing over in prophase 1. Random orientation is also really important, both in metaphase 1 and metaphase 2, because the random orientation of the homologous chromosomes means that there is 2 to the n possible orientations in metaphase 1 and metaphase 2. That's 2 to the 23rd in humans, or 8.3 million different combinations in the gametes. All right, so that means that this is, chromosome 1 is here at the top and chromosome 2 is here at the bottom, but they could very easily switch. With 2, that's not so important, but with 23 or 46 total chromosomes, there's all sorts of possible combinations that you could include there. Same with metaphase 2. So chromosome crossing over and random orientation occurs during meiosis and effectively creates an infinite genetic variation in the haploid gamete. So that's pretty cool. You could practice thinking about this in a minute, but which part of the process, meiosis 1 or meiosis 2, produces the most genetic variation? And give some evidence to support your answer. All right, 3.3S1, drawing diagrams to show the stages of meiosis. We're not going to worry about that yet. We're going to come up to it here in a second. But the biggest one here that you wanted to talk about before we get to the S1 is U4, which is the early stages of meiosis that involve pairings of homologous chromosomes, and then, of course, the crossing over that I just discussed. So in prophase 1, the homologous chromosomes that are associated with each other get together to form bivalents, or synapses. Synapsis is singular, synapses is plural. So bivalents are basically homologous chromosomes, all right? So the DNA is going to supercoil, the chromosomes are going to condense, the nuclear membrane is going to dissolve, centrioles are going to migrate to the poles of the cell, and crossing over between non-sister chromatids is going to take place during prophase 1. And this is where we get the recombination that we just discussed. It's a big source of genetic variation, but the pairings of the homologous chromosomes is really important. They got to stick together through meiosis 1. And so that occurs at prophase 1 through the process of basically getting super close to each other and creating a bivalent. Okay? All right, U5 is about the orientation of the pairs of homologous chromosomes. It's random. So the bivalents will line up at the equator, but in any possible combination. And again, I repeat the fact that this is figured out through 2 to the n. Remember, n is the haploid number of chromosomes. So 2 to the 23rd in humans is 8.3 different combinations for the way that the bivalents could line up along the metaphase plate. Okay, here's anaphase 1. You can pause it. I'm not going to read over each one of these. Here's telophase 1. Pause it if you want, or go to the slides by themselves. Prophase 2, just looking at the pictures. Metaphase 2, anaphase 2, telophase 2. Okay, so I don't want to review every single step of that. We did it in class. You did it as homework. We went over it again. So if you need to review this, watch some videos, practice drawing it, practice drawing it. But you should be able to draw each step of this, thinking about what's happening with the nuclear membrane, what's happening with the chromosomes. Okay? Try to do this for every phase for meiosis 1 and meiosis 2. Let's go to A3, which is actually A2 and A3. So this is about description of the methods used to obtain cells for karyotypes, and A2 is looking at the fact that aging influences the chances of non-disjunction. So this is pretty straightforward. The risk of having a child that has Down syndrome increases very greatly as the mother gets older. So oftentimes it's advisable for mothers in a high-risk category to be chosen to have prenatal testing, which just means before birth. Okay, you can see that blue is risk of trisomy 21 or Down syndrome. Red is risk for all the trisomies, so 21, 13, 18, any X or Y trisomies that could occur. The two ways that you can do this to find out, to get like karyotyping analysis done, is through amniocentesis or chorionic villus sampling. Those are the two ways. Should you know those two ways? Absolutely. So data from a positive test can be used to decide the best course of action. For some parents, just to be aware, that can mean abortion. It can mean adoption if they're not ready, and it can also, of course, mean keeping the child and just recognizing the issues that are going to come from that. And that is often the case, and it just gives parents some time to plan and figure stuff out. One of the really interesting things that I thought was really cool, and I didn't know about this till I started doing some research on this, is what's called the nuchal translucency scan. So it's a non-invasive test that's carried out between 11 and 13 weeks, where when they get their ultrasound, they look at the nuchal fluid behind the neck of the fetus. You can kind of see that here. So this is the baby's head, and here is the neck part. If there is a large amount of fluid, and you can see that by the black here, it's strongly correlated with Down syndrome. So oftentimes if the doctor sees this and sees a lot of nuchal fluid, they will advise them going through amniocentesis or chorionic villus sampling to confirm. They don't just do this and say, oh, yep, your baby has Down syndrome. They do this first and then use that as a great guide to figure out whether they need to do the next step. All right? You can watch a little video here and see whether or not you could identify the scan as being positive or negative if you wanted to practice. Here's amniocentesis. This is where the amniotic fluid is taken via syringe. The needle is guided in using an ultrasound to make sure that the baby doesn't get poked or anything. The amniotic fluid has cells from the fetus, and so they use those cells. This is usually carried out in the 16th week of pregnancy. There's about a 1% chance of miscarriage due to amniocentesis, so it's a pretty low risk. Chorionic villus sampling is the other way, and the reason that you would pick one or the other is chorionic villus sampling, it can be done much earlier, although there is about double the chance of miscarriage. So the chorion is the membrane, which is a part of the placenta. It contains lots and lots of cells, and so those can be taken and then cultured. Right, villi, do you remember? Those are the little fingers. So all those cells are going to have lots of information. So once they get lots of cells, they go and they culture them and they grow them, then they're able to carry out the karyotype. And again, this can be done around the 11th week of pregnancy, but does have a 2% miscarriage risk. We already talked about this, but just a reminder, risk increases as mothers get older. There's a little discussion here if you want to do that. Okay, that's it for 3.0.